Okay. So now we are going to talk about the another database which is called string. Okay. So if we do the network analysis, enrichment, interpretation, and also we are going to talk about what are the common mistakes that the people do. Okay. And because we know that a gene or protein do not work on its own. Okay, it works together with some other proteins or some other genes. Okay, that may be transformed into a disease. So in a drug discovery, what happens is some people say that I'm working on this particular protein. Of course, you can inhibit it or you can activate it. Suppose if you do not know that whether how this protein is interacting with other proteins. Alright? For example, you take the protein X, it is also associated with A, B, and C. If you inhibit the protein A, X, it will also inhibit all these three proteins. And that's why it may cause some kind of a side effects or adverse events. Okay, so that's why it is called as the network analysis. So you need to use knowledge to make a decision whether if I work on this particular protein, okay, so what are the possible side effects, okay, or adverse events. Those things actually you can get from this string database, okay. Okay, strings help researchers move from a single target to a pathway-informed drug discovery hypothesis by visualizing known and predicted protein associations. Okay. Now, from this one, suppose can we say that Arjun, since you say that can I put a title that EGFR my hypothesis is so and so compound is going to work on EGFR mutant. That is not acceptable because it is telling only a partial story. In yesterday, we have seen that this EGFR, they have a three different pathways. Okay, if you look at the slides. So now you need to say which pathway, specific pathway. Okay, that one is okay. And why this matters is targets rarely act alone. That's why I already told you. Always stick together. Cancer signaling is a network based. Of course, if anyone looks at the cancer one, really they'll get a lots of headache. Resistance may arise through bypass pathways. Okay. And here, in general, what happens is in any network, there are two things. One thing is called as upstream regulators. Then the other thing is called downstream mediators. These two are the terminologies that the people use in drug discovery. Okay. Upstream modulators means which is in the network pathway, which is at the top level. So when you work, your compound works on this, am I right? That is called upstream. They are called as a regulators because they are going to regulate all these pathways. Then the other one below this one, there will be some other genes or protein. They are the ones that they produce a response. They will call it as downstream mediators. Okay. So which one is good? Whether we should stop here or we should work at the bottom one. Okay. Suppose if you work at the bottom one, okay, let us say X is the A, B, C. So you are working on A, then still the disease may progress from B or C. Still. Am I right? So that is the problem. That means your compound may not effectively cure the disease. Okay. Then, but if you upstream one, if you close down, definitely A will stop, B will also stop, C will also stop. But in that one, C is the one that is very important for us. C activation must be there. So if you inhibit C also will stop, that means we'll get some kind of a side effect. That is the reason why you see the nausea, vomiting, diarrhea, all other things, muscle weakness, everything that you will get, because all these are found to be very effective. That's why the people say that this is a breakthrough medicine. Why they call it is a breakthrough medicine? It is working on the upstream regulator. That's why it is able to do this one. So disease is going to be subsided. Then all other things are the people who take those medicines will suffer. Maybe with diarrhea, with nausea, vomiting, okay, headache and all these things. But what these companies say that, oh, all these are manageable. Forget about it. Okay. But they will sell it as a blockbuster drug. Okay. Then because they say that because of nausea, no one will die. Because of the vomiting, no one will die. So that's what they say. But the patients, they don't want. Okay, that's why in a cancer treatment, even though it is progressing so much, but many people are not willing to take chemotherapy. Because I know many people, they say that yes, I'm suffering with the cancer. Okay, because if I take chemotherapy, I lose my status in the society. So if I work, if I live for 2 years, that's fine for me. Okay, and rather than taking this chemotherapy and telling to the entire public saying that I am a cancer patient. Okay, the people don't want that. Social stigma is very, very important. Okay, and resistance may arise through bypass pathways. Combination strategies need pathway rationale. Okay, this is the very important point. Now you look at the point. Am I right? X, A, B, C. Already I told you if you inhibit X, what is the disadvantage? And if you inhibit only one of the pathways A, what would be the disadvantage? Now, yes, upstream regulator is good, but it's also dangerous. That's why now you need to look at A, B, C. C is good. You don't want to this one. But A, B, you want to inhibit. Then you need to use combination. Okay. That's why we cannot even simply think that to treat a disease only one single medicine works. It may not. It may not. So whether in our drug discovery proposal, whether we need to discover more than one medicine to treat disease completely. For that, we need to know this network analysis. That's where it helps. That's why you will see the combination strategies now. Okay, EGFR interacts with this family of proteins, and these are adapter proteins activated in MAPK signaling. Then just now I told you that there is a mechanism pathway that PK3/AKT supports survival. Okay, our network context explains response and resistance. Okay, here use string to ask better drug discovery questions, that not merely to generate a colorful network image. Okay, of course, when you do this one, it comes with a very colorful image. Okay, string is a hypothesis generation tool for pathway informed discovery decisions. Okay, even though many people talk about this one, still now the biggest challenge is inflammation. Inflammation causes so many things. Everywhere there is an inflammation. Until now, there is no single drug, okay, that works. That's why if you go to a doctor, he will prescribe all the medicines. Apart that, they will also prescribe paracetamol or something else. There is something. Madam, next slide. Okay. This one. Here, again, you look at this validated target. This is the network content, which proteins communicated with this one, and pathway. Then you need to look at the co-target class, which bypass mechanism may matter. That means this is where co-target means whether you require a combination therapy or a monotherapy, or what is the next step. Okay, structure, bioactivity, and candidate design. So what it will do is, because here this is the key message, for each and every database or whatever the thing that I'm talking about what is the thing that is good and what is the thing that is not so good. Okay, what does it mean? That means every database or every AI tool has one positive and also has negative thing. Okay, so you need to be aware of the negative things and you should encash the positive things that are associated with it. Okay, So, which pathways are overrepresented, which interactions have a stronger evidence, which co-target hypotheses are plausible. Of course, if you do the practice that you will get it, okay. String does not directly prove direct physical binding for every edge, it won't tell you. And cancer cell specific interaction activity also it won't tell you, because you are talking about EGFR. EGFR may be present in any, also may be present in other diseases. Okay, but this network analysis won't tell you only about cancer. Okay, and drug, it won't tell you whether drug is going to be effective or not, and correctness of every predicted relationship. Again, you have to go back and check. Okay, and best mindset is use string to generate a biological map, then validate with literature and omics data and experiments. Okay, there's a lot of. A network is useful only when it changes your biological reasoning for next experiment. Okay, next one. Now here, what is the core output that you will get is you will get a protein-protein association network, and you also get the confidence codes for edges, and you will see what is the edge, and evidence channels, and direct and indirect functional associations. So here function means the one that we see. Okay, so direct means when the receptor is activated, automatically it produces a biological response. That is called as a direct. Indirect means when you alter that one, then it will go through some other protein, then only it will produce. That is indirect ones. Okay, and the indirect ones is bit difficult. Okay, and it is called network clustering and functional enrichment analysis. This is a completely different technique. Okay, and here you can say that experimental data, curated pathway, co-expression, gene neighborhood, text mining, and also we can also do the computational predictions. Okay, whatever you output you get, you need to do these things. Okay. And where it helps is it identifies the network context, helps explain the resistance, and suggests co-targets. Okay, it also supports mechanism figures, and whatever the figures that it gives, even it's very difficult for us to draw. Even that itself is an AI tool. Okay, AI tool is already embedded in the string. Okay, because in the gene cards we have seen one human skeleton, am I right? In which there are red dots. It is AI. It's not a single static image. Okay, so now drug discovery value and supports mechanism and prioritizes follow by. String integrates multiple evidence types, therefore evidence inspection is essential. Okay. Next one. Now, this is the beginner question. What you need to do? What proteins are connected to EGFR? Previously, you checked for only EGFR. Now you are seeing that what are the proteins that are connected with EGFR? Because in drug discovery, you talk about the selectivity, you talk about the toxicity, and all other things, right? So, for you to understand that one, you need to know that which other proteins they are associated with it. So, what are the nodes and edges? Which pathway names appear? Can I export the network image? Yes, definitely you can export the network image. Okay. And drug discovery question is what interactions explain EGFR driven growth, especially in the case of cancer? Then what are the pathways and which partners are possible and what interactions deserve validations? Okay. So here I want to say this one. Okay, this is one of the common mistakes what the people do, okay, in an academic research. When you say, I'm taking a simple example of cancer, okay, they take particular thing, they ask they culture the cells in a plate and they will put it will be killed, and they say that they have anti-cancer effect. It's not correct. It's not at all correct, because anti-cancer, there are three different terminologies that the people use: cytotoxicity, anti-cancer, anti-tumor. Okay. Cytotoxicity means whatever we do in vitro, that is called cytotoxicity. Strictly speaking, we cannot use the word anti-cancer for that. Anti-cancer means if you use in an animal model, it shows the effect, and we should call it as an anti-cancer. Okay. Then if the compound shows the activity in a human, it is called as an anti-tumor. Okay. And many people do not know, even I also do not know when I started my research. Okay. So actually I started my research when I moved to Malaysia on cancer research, because I was so passionate, as everyone thinks that the cancer research is the greatest, like that I also started. Okay. Then I got lots of compounds, everything. That time, the system came with a 3D culture. 3D culture. Okay, that is the most appropriate thing to check. So I used to say so great about it, and the people also used to think that Arjun is doing a great cancer research work those days. Okay. Then I also thought that I'm doing a great job. Then I got one person in UK, and those wife and husband, they started a contract research organization company, and they said that Arjun, now we come up with a new thing, because in 2D cells, what happens is these cells are already clustered, they're competing with each other. And I know the lots of tips and tricks. Even the compound is not anti-cancer, I can show the experimental proof it is anti-cancer. I can lots of tips and tricks are there. I don't put the nutrients. That's all. The cells will die. That's all. Oh, the cells die because of my drug. Yeah, lots of things can be done. Okay, but in reality, that is not there. Am I right? And actually, you are sticking onto it. You are sticking onto something. It is called as the adherent cells. Okay, they're sticking onto it. Okay, so then they come up with a 3D culture. When I put the compound into a 3D culture, they did, and they said that sorry, Arjun, none of your compounds are active. All my 7 years work has gone. Then I fed up with the cancer. There is no way that I can do it. Okay. Then I fed up with it and listen. Because why I'm telling all these things is, okay, of course, when you are young, yes, you can use the words. For everything, you can use the word anti-cancer. But when you are growing up, actually, you need to understand what are the differences. Okay. So, uh, yeah, that's why what interaction explains what pathway branches where I'm talking about. EGFR. Drug discovery question. This question, right? Okay. Which EGFR associated proteins and enriched pathways support a combination strategy for EGFR TKI resistance in NCLC? You see, previously what I say, only EGFR. That's all. Am I right? Now I am going little bit advanced. You don't consider not only EGFR, you also should look at other proteins, okay, which are closely associated with EGFR, because your target was not to, your end strategy was not to synthesize very fantastic molecule that works only on EGFR, okay. Your strategy, your aim is you want to treat the cancer, and you know that EGFR do not act alone. It also acts with other proteins, so you need to put them together. Okay, so that's what we are doing. That's why the string database is going to come. Okay, this question guides settings, interpretation, and reporting. Okay, do not start with the network image, start with the decision the network should support. Next one. Okay, and a reproducible approach. Actually, when you see this one, I always use the word reproducible because it has to be this one. Okay, then search the correct protein. So you have to use the protein name, gene symbol, or accession. I think my colleagues say that you should use UniProt number. Okay, and select organism carefully. Sometimes the people don't select the Homo sapiens, that is human. Okay, start with the default network, don't make any changes. Then afterwards, if you want to play around, you can adjust the settings. Then these are the KEGG and Go and KEGG or Reactome. These are the very powerful pathways, very powerful pathways. Okay, and which is very difficult for me, which is difficult for anyone to understand. And actually, these ones, I will ask my team to explain to me. Okay, it's very difficult for me to understand this. Then you have to export and document it. Okay. Run the same EGFR steps twice, first with a medium confidence, then with a high confidence. Of course, you can check it. This demonstrates that settings are not cosmetic, they directly affect scientific interpretation. Okay. Okay, reproducibility requires regarding both the query and network settings. Next one. Now, this is what you are trying to get. These are the different images very beautiful images and all those things will occur. Okay, because actually these kind of beautiful images will also help to succeed in getting the publications. Okay, and also in successfully obtaining the research grants. But however, whatever the image that you put, you must be in a position to explain. Okay, then the problem will start. Okay, node means a protein or gene product. Okay, edge, previously, yeah, edge means association between the proteins. Okay, when you put the more edges, then you say more connected network. And if the line is thicker, that means they are closely associated. And the clusters, that means when you see them together, then they say that with the related functions or pathways. Okay. Nowadays, the people are talking about AIA and all those things, but these databases are there very long time back. Okay, maybe 15 years back itself is there. But those days, the people never used the word called artificial intelligence. But nowadays, the artificial intelligence became a fancy term. Okay, but all these are really there in the past also. Okay? Now, the scientific interpretation asks whether each relationship is biologically plausible. Again, how you check that one is you look at the literature, and it is supported by a strong evidence, and it is relevant to the cancer, and it should be related to the proliferation, survival, or resistance. Okay. In cancer research, we need to know the differences between these three terminologies: proliferation, survival, or resistance. And here also, most of the people who do the research, okay, in cancer, this is the common mistake that they do. What they do is they put the cells, okay, they allow the cells to grow, and in another compartment, they will put a drug, it will allow to grow. Maybe 24 hours, 48 hours, or 72 hours, it depends upon that. Afterwards, they will compare these two. And they say that the cell count here is less compared to the untreatment. But from that, how can we say that it is killing the cells? Actually, it is preventing the growth of the cells. It is preventing the growth of the cells. Okay, that is called as the proliferation. Okay, that's why, strictly speaking, it is called as an anti-proliferative effect of the compound. Am I right? Because in the treatment, what we want the compound to kill the cells. It's not to prevent the growth of the cells. Am I right? Okay, that is also required. It also should kill. And most of the experimental protocols that they do in anti-cancer research, they do only anti-proliferative effect. Actually, they don't do other things. Of course, that itself is a big topic. The another one is a survival. Survival means you put the tree compound, some cells will die. Okay, let us say we say 70% of the cells die. Then we are not telling why 30% is surviving. Am I right? Remaining 30% is really surviving, and no one talks about it. Then we talk about the resistance. Okay, so when you treat. Okay, this also I will tell you, because cancer cells is one of the difficult to procure ones. Okay, and most of the laboratories, what they do is whatever the cell that is available, they will get it and they do it, and they do not know what is passage number. There is something called a passage number. Okay, a passage number means when I take out from the supplier, I will put into the nitrogen one, that is the first passage. Then afterwards, I do a subculturing, that passage 2, passage 3. Okay, and strictly speaking, from passage 4 to the passage 10, only we should use, and apart from that, we should not use. And most of the papers most of the students who present the anti-cancer effect, the first question that I ask is, could you please tell me what is the passage number of the cells that you use? And if they do not answer it, then I say, okay, you just proceed, but your results are not reliable and repeat this one. Okay, those are the things that we need to be quick. Okay. And because why the passage number 10, this one means because it develops the resistance. That's why passage number 4 to passage number 10 is the most important. Then you may ask, why can't we use passage number 1 and passage number 2? The reason for that is, actually when the cells are shipped, they are shipped under ice. Okay, and usually at -80. And then afterwards you are putting into the nitrogen cylinder. Okay, then all these things you need to slowly bring it down to the room temperature. That's why it requires process 1, process 2, process 3, and process 10. Yeah, that's the thing. So the network is a map of thinking. It is not the final biological truth. Better next one. Yeah, no, next one. Okay. Here, this is what I said. Am I right? Because this kind of a word confidence you will find in lots of the databases. Okay. More medium confidence, high confidence, and very high confidence. Okay. Here you can see that when you put a medium, you will get a more notes, initial exploration and brainstorming. Okay. Just look at the picture and think, but don't do anything with it. And high confidence, when you are going high, your notes, your this thing will appear. The image will become lesser numbers. Right? So this is useful for the reporting. Okay. Then very high confidence, you may fail. Okay. Then more interactors. Okay. Then you can see more things, and fewer interactors, you may miss out something. And evidence reveals why an object exists, but again you need to look at the scientific interpretation. Okay. Because here again towards the end in drug discovery research, you are the boss. Okay. Don't give that role to any research assistant. You are the boss. You make a decision. You make a decision whether you want 0.7 or 0.6 or 0.5. Okay, that's very important. Okay. Higher confidence improves reliability but may reduce biological knowledge. Okay. The next one. So what enrichment asks is, are the proteins in my network overrepresented in a specific biological functions or pathways compared to with what would be expected by a chance? Okay. That means, let us say if you take out the inflammatory pathway, it is also there in cancer, it is also there in diabetes, it is also there in other diseases. Okay. Actually, in enrichment, actually it will tell you which disease it is more predominant. It is more predominant. So, for example, I can tell you how many of you know that metformin is also useful to reduce the weight. How many of you know? Yeah, it is one of the best drugs that the people think that it is good for the weight loss. Okay, yeah. So why means if you look at that entire network, actually you will understand. Otherwise, the people think that metformin is only anti-diabetic. Okay. So of course now recently because of nitrosamines and all those things, there has a problem. Okay. So why I give that example is because the way that metformin works, am I right? The highest enrichment is there in the diabetes, and the second enrichment is something related to obesity. That's why it is also helpful. Okay, so that's why I tell that example. Okay, so what to look for? You have to look for. I told you about the pathway. EGFR, ERPB signaling, MAPK pathway, PI3, AKT. Of course, there are so many pathways and receptor tyrosine kinase activity. Okay. Use enrichment to write a concise pathway rationale. That means, okay, when we started this thing, we started with a cancer. Okay, lung cancer. Then we narrow it down to lung cancer. From lung cancer to, we narrow it down to the non-small cell lung cancer. From there, we come to the EGFR. From the EGFR, we come to the EGFR mutant. From the EGFR mutant, we come up with a pathway. So this is how you are narrowing down the discovery question. Okay. Enrichment converts network graphics into a mechanistic drug discovery argument. Next one. Okay, and what to search and how to structure it, you need to look at the target context. Okay, and pathway mode, select multiple proteins. So this is focused ones and candidate co-targets and expand interactors, inspect evidence. Okay, and omics interpretation, input a gene list of from transcriptomics or proteomics. Okay, this is the one. Am I right? Here I want to take something. How this one, how I use this network, okay, to perform the experiment. Am I right? So in the EGFR network, when you do, you will get lots of pathways, right? And I will check, let us say X, A, B, C. Am I right? So my experimental protocol, my first experimental protocol is on X. Okay, I will check it. So whether it works, it's preliminarily passed. Then I check this A, that my second experiment is A. Okay, then I do for B, I also do for C. Then afterwards, I go look at whether it is anti-inflammatory effect or not. This is called as a complete discovery proposal, discovery experimental protocol. You cannot say that some people will work only on this one, I discovered something, or some people will do at the bottom one, they will say this one. So complete discovery means you are coming from the upstream regulator, downstream mediator, and the effect. And for each and every experiment, there are two, what is that called as, there are two experimental protocols. One is predictory or informative, the another one is a confirmatory protocol. Okay, that is always important. You cannot confirm the activity of the compound by a single experiment. Okay, because why I'm stressing so much is in my initial career, my research proposals were rejected so many times. Then when I inspect, then I know what mistake that I have been doing at that point of time. Okay, and drug mechanism figure, mechanistic illustration for reports, and evidence grounded AI use. Okay, this one is whatever you get, actually you can use any AI one. Okay, do the things, look at the database, put that input into the AI, then you look at the output, again you confirm it. Don't work in the other way. That means never ever start your research with your research assistant. Okay, first to the database, then afterwards you can give to AI, then you will get the fantastic proposals. Okay, the best string search depends on whether you are exporting a target, a pathway, or an omics use. Next one. Okay, practical habits that improve the scientific quality. Always confirm the identity. Okay, for EGFR, do not accidentally analyze a non-human protein. Okay, inspect evidence. Okay, whether support comes from the experiments, curated databases, co-expression, prediction, or text mining. Okay, and we should always believe the experimental data only. Okay, not others, and keep it readable. Okay, use high confidence, limit interactions, export the figure, write it. Okay, good string analysis is selective, documented, and biologically interpreted. Next one. Yeah, avoiding misleading network conclusions, and these are the common mistakes. What the people do is this one. Okay, and these are the better practices, and these are the problems that are associated with this one. Okay, and the biggest mistake is treating string output as proof rather than evidence to interpret. Because nowadays what the people are saying is whatever it come, okay, like it should be right. That means we are thinking that the other researcher who developed the string is much better than us. Okay, next one. Okay, and here also very similar. String show that EGFR interacts with many proteins. That is that's not correct. Actually, I would say that a string network centered on a human EGFR. Okay, the thing is here you are saying that which database you are talking about, the human. Okay, was generated using a high confidence interaction threshold. The network was enriched with ERBB, MAPK, and PI3K AKT signaling, supporting EGFR as a pathway connected driver of proliferation and survival. Okay, this is what you are going to tell. Okay, then report the method, result, interpretation, and the limitation to the plan. Okay, always you need to write the limitations. Okay, next. Okay. So co-target should be chosen to solve a defined resistance or pathway bypass problems. I told you that simply our discovery is, our intention is not to discover a fantastic molecule against a target. This one is to treat the disease. Okay. So these are the potentially easier for relevant partners. All these are the pathways. Okay, how to use this type of, okay. Do not immediately claim a combination therapy and instead ask, is the co-target active in my disease model? Is there biomarker evidence? Is the combination tolerable? Can I test it experimentally? This is the very important thing. Everything, whatever you do, and you should be able to do it experimentally. Okay, next one. Yeah, these are the take-home messages. Okay, string places a target inside a biological network. Interactions can be physical, enrichment can be done. The next step is literature, omics, or experimental validation. Okay, do not use string to decorate a project. Okay, please that one. Okay, next one is, I think this is the last one. Last one, right, madam?
Yes, sir.
Okay. Can you open that one? What is that? Okay. What I do is, okay, just go through this one. Yeah, this is the string database. Okay, there is a step-by-step procedure is there. Okay, next one. Do only these things. Okay, again, I am saying that if I want to explain all these things, it is going to take a very, very long time. Just read, do it slowly. Okay, do it day by day. Sorry, on this one. Okay, next one. Next please. Yeah, you will get a very, in general, am I right? In higher impact factor publications, actually, you can see this kind of a network, okay? And actually, it improves the chances of the paper getting published, okay? Especially those who are writing review articles, these kind of images really help, okay? And nowadays, since I'm talking about the images, the success of your project or the success of your paper acceptance, it entirely depends upon the image. Okay. So usually my strategy is before we write any paper, we ask my team to generate 3 images first. Then if I'm satisfied with those 3 images, then only we go and starting writing a paper. We don't write a paper first. We write because without images, no one is going to accept. Okay, and why they don't accept, whether it is right or wrong, of course, I can talk about it for about half an hour, why that trend is happening. Okay, these are the things, and here also they say that use more options, do this. Next one, madam. Yeah, 7, 8, next, yeah, you will get so much this one, and next one, yeah, and just you record your these things here. Okay, so that is about this one. Of course, these are some of the tips that we do. Okay, so can you open strings database? Yeah, put strings there. So yes. Yeah. Okay. Here. Okay. You can go to search. Actually, big people, what they do is here you can put easier for. Actually, here you can see that the string chart new model. This is the AI. Okay, this is the AI. Okay. Yeah. Okay. So in that one you can search. Yeah, here you can play around. Here, if you see this one, am I right? But can you? It's good actually. Of course, different colors indicate different things. Okay, thickness indicates lots of things. Actually, okay. Now, okay, this is what we can use it. Now, if you see this one, you are getting confused, am I right? Okay. What you can do is you can copy this image, put it into the generative AI. Can you please explain this image in a layman terms? And it will explain.
Layman.
That's where you can use your research assistant. And from that, once you understand that, hey, what you are talking about this CVL, I did not understand what is it CVL. It will tell. Even if you say, hey, you are telling me in English, can you tell me in Telugu? That also will tell. Or it also tell, no, I don't have a habit of reading. Can you speak for me? It will speak for you. That's where the AI will help. That's how we should use AI. But you should not use AI to generate this map. That's not responsible use. Okay. So then go down a little bit. Here you can see that, of course, legend, settings, analysis, exports, clusters. There's a lot of things that you can do. Node colors. What are the agents? Can you do? What are the known interactions? These things. Okay. And actually, interpreting this data itself is the biggest thing. This is the way, here you can see the confidence scores. Usually we go at 0. Up to 0.7. Even after you do that one also, okay, you put this one, entire thing you can copy and put it into a generative AI. Can you tell, oh, now basing on this one, I want to develop a hypothesis. Can you help me to develop a hypothesis for me? It will develop. Okay. Once it develops, never ever blindly accept whatever AI says. Whatever AI says. Okay. It can bring you to the very high level, or it can bring you down to the very low level. Okay, please. That's why don't put so much hopes on AI. Of course, when I talk about AlphaFold, I will tell you why is it so. Okay, so this is about this one. Okay, I think what we do is we try to do one more lecture AlphaFold, and afterwards we take it. Is it okay? Okay, it seems the participants are good. At least I don't see anyone sleeping. Okay, that is good thing. And the second one is not making a noise. Okay, and of course, sometimes while opening eyes also we can sleep. Okay, that is called as a brain sleeping. Usually it happens. Okay, but it's okay. Okay, now here this alpha fold. This is the one thing actually that is going to take away. The people are getting worried in a drug discovery research. It is going to take away lots of people out of the jobs in drug discovery. Okay, so no one knows whether it is good, whether it is a good or whether it is not good. The people who are fancy about this fancy term artificial intelligence, they are appreciating. But the people who are really struggled, worked hard in research, and developed some kind of expertise, spent about 20 to 30 years, and they are thinking that, oh, they are going to become useless. Okay, and this AlphaFold, the what is it? Who has introduced this? Okay, he got the Nobel Laureate Prize. Okay, and it is a biggest surprise to the entire scientific community in the world. Okay, how come a particular scientist is being given a Nobel Laureate Prize, okay, that does not have any experimental evidence. Until now, all the Nobel laureates Okay, especially in this one, only for the experimental work. Okay, but this is the guy. There are lots of things can be learned from this guy once. So there are both pros and cons. The people are talking a lot about it, but the message that I got from here, from this guy, is basically he's a biochemist. He knows the subject. Okay, then he's making use of the research assistant. That's why he won it. That's why he became more successful. Okay, what I mean is, you are a pharmacy or you are doing a biological sciences research. That is your core expertise. Try to master it first, then go for the AI, how it is going to help you. Don't waste your time to master AI. Okay? Try to develop this knowledge, then whatever the things that you got, this one. Okay. Then I also told one word that saying that why some people are, of course, this was happened only in my recent visit to UK when we are discussing actually, uh, you know, in a designing molecule, what we need a protein, and we need to know it's a 3D structure. Once we know the 3D structure, then we need to see whether our compound is fitting in or not. That is entire about computational aided drug discovery. That's all, nothing else. Am I right? So to get this protein, the people used to do X-ray crystallography, okay, cryo-electron microscopy. Even they also use NMR. Okay, they used to take a very long time. Am I right? And that is called as the structural biology. And that structural biology is one of the most important key concept in a drug discovery. Because if you do not know the structure of the protein, there is no way that you can design the molecule. Am I right? So then when these people have said this one, they say that we are going to lose all our jobs. Because this guy is trying to predict a lot. Okay, so that's why even I'm struggling with one protein for my research work. Then why I asked them, they say that, hey, why can't you try with alpha-fold? Then I say, how? Oh, why you are saying that one? So what to do? That is the reality. Am I right? So, and this is one of the best examples where you need to say that is we need to stick to our grounds first. Okay, don't try to jump one to one to one to one, and Rolling Stones gather no mass always. Okay, that's it. Okay. Now, why this matters for the EGFR mutant? This one is protein structure is the bridge between target biology and molecular design. That's what I told you. AlphaFold DB gives rapid access to the AI predicted protein structures and confidence metrics. Okay. Again, I told you confidence is different. It means, right? For drug discovery, the value is not simply viewing a structure; it is knowing what can and cannot be trusted. Okay. Then, of course, this one we are talking about. Can we use the predicted EGFR structure safely? The key word is safely. To understand domains, mutations, and possible binding site context before moving to the experimental structures and ligand data. Okay, and you see, AlphaFold is a powerful starting point, but not a final drug design answer. Okay. Here I want to tell that I think I have written my first AI paper, I think during COVID time, okay, in a drug discovery today. Okay. Since we cannot go to the lab, so all the labs are closed, then my team asked me, Arjun, anyway, you won't keep quiet. We have to do something. Okay. Anyway, someone is talking about AI. Why can't we talk about AI? What is its real use in drug discovery? That is the first paper that we published in the drug discovery today in 2019 or 2019. Okay. So at that point, from that point until now, everyone is saying that they throw so much. Almost 8 years, 9 years have gone. Can you tell me, is there any product that came from the air which is there in the clinic? Mhm. Not at clear the clinical tracks. Still at the FDA, no, until now there is no single drug There is no single medicine which is approved by the US so far. And because I met few of the, I'm not getting the names, who are started up this company, startup companies of AI in drug discovery. I'm forgetting his name. Uh, he is from the University of Dundee product, and I'm not getting the name of the company. So straight away he says so many things. Oh, that so many drugs reach to the clinical trials and all those things. And later on, no one knows what happened to it. Okay. So, sorry. Not even safety, even efficacy also it is not passing. Even it is not passing phase 1 clinical trial. Okay. Not at this one. So here I will tell you, okay, why. This is my gut feeling is why AI is not working for drug discovery. Unfortunately, it is because of the fake data that is available in the literature. Fake data available in the literature. Someone say that the anti-cancer of activity, IC50 activity of this compound is 10 nanomolar. The another one say that 10 micromolar. The another one say 100 nanomolar. He doesn't know. Now that is the biggest problem now. That is really the biggest problem. Okay. Of course, later on when I'm going to talk about Campbell, then I will tell you, you will see that for the same drug, you will see the different values for the same target. Okay. That's why it is, that is also could not able to do. That's why someone has asked me, Arjun, if AI really to be successful, okay, in drug discovery, what should we do? One thing what we should do is we should remove all the literature that is existing in the world now. Hibernate it. Because all that is a fake data. Everything is a fake. Everything is a real, even including patents is also fake. Okay, when I'm saying that 70 to 80% of the things are fake. When I say fake, it may not be intentional. Okay, it may be an experimental error and all these things. That's why, AI, when I put myself, it will recognize, oh, this is Arjun. Because, but if I do a cosmetic changes and go, then it may not recognize me. Am I right? In the research also, most of this thing is a cosmetic things because it's to write a paper. The goal is again the problem is writing a research paper and completing a PhD thesis or completing a MPhil thesis or anything MSc thesis. So there are lots of things like which I do not want to go into that one, but that is my personal view. It is nothing related to my university's view. So and then okay, AlphaFold is this one. Start with a scientific question and not with a website. This is what I have been telling so many times. Okay, so that it will impress upon us. Next one. Okay, target validated, sequence identified, alpha model, confidence check. Of course, if you are lucky, you can check the PDB because all the PDB is the experimental design points, and design hypothesis. Of course, my next lecture I'm going to talk about PDB. I will also tell you what happens. What AlphaFold helps answer? What is the likely 3D fold of the protein? The important is likely. It is not saying that the absolute 3D structure. It says that how likely it is going to look like. Okay, which domains are structured and which may be flexible. It will tell you. Okay, is a region suitable for structural interpretation? Yes, it can tell you. Which residues or domains should be investigated further? Okay, it cannot tell whether ligand will bind to it or not. It usually lacks bound drugs, cofactors, and disease-specific context. Because I told you that in a string database, you say that for one protein, that protein will be active only in the presence of another protein. So you need to crystallize these two together. Am I right? Then only you can see that confirmation because Suppose if you see, take a one protein, it is like this, am I right? So it looks like this. When I'm putting another protein nearer to it may flip like this. The conformation may change. The shape may change. When the shape changes, your drug may not fit in. So that one alpha cannot do. It does not replace PDB structures. It does not remove the need for experimental validation. Okay, but even without that, also we got it. Next one. So what the database provides is a pre-computed protein structure predictions generated by using AlphaFold methods. Okay, and search by protein name, 3D viewer, confidence merits. Okay, and here the good thing is it's a rapid. You whatever the protein that you are working on, actually you can use this. Okay, here I want to tell my own personal experience. Okay, actually, at this point of time, I'm working on one project in which I'm going to synthesize a new molecule working on Candida, multi-drug resistant Candida auris. Okay. Until now, there is only one drug molecule which is there in the market, that is by Pfizer. Okay, that is called Manogenix. Okay, but still it is in the phase 3 clinical trial, but it is being used for accelerated use. Okay, so when I did that, I started with a great enthusiasm that I think I'm working on a particular enzyme called GWT1. So then I said that is the first time in my career why I cannot synthesize the GWT1 enzyme myself. Okay, because GWT1 is not at all there. That protein, no one knows how that protein is there. Then I made a bold step to see that whether I can synthesize the GWT1 protein itself on my own. Okay, so on my own means my research team, but by taking the others' help, we did all the things. We could able to get a GWT1. Am I right? Then, but we could not able to get in a pure form. Am I right? So ultimately, again, AlphaFold is one that helped me. Okay, then go to the GWT1 predicted structure. Okay, so of course, whatever the compounds that I got, only 20% showing activity, remaining 80% is not showing activity. Okay, so in that way, that means there is something in it in AlphaFold, but it is not always because without this AlphaFold, I might have not carried out that particular project because there is no protein structure. There is a so in that way it will help. Okay, that's why it is called as a pre-computed protein structure. Okay, and all these things. Okay, and rapid structural starting point. Okay, well, when no experimental structure is available, that is my example. Just I told you that supports domain mapping. Okay, helps plan which experimental structures or assets are needed next. Useful for teaching structure function relationship in targets such as next one. And this recommended search flow. Okay, please this one among all these databases, UniProt is UniProt will give a code. Okay, and that code you should use. That is the most ideal way of doing the things. Okay, and check protein length and domain descriptions, download or record alpha entry, record the exact accession. Okay, and use identifiers carried forward from the gene cards or UniProt rather than only common names. That means all these are linked to one each other. Right? So search for this one, family proteins, same Homo sapiens. Okay. Search by accession, confirm by biology, then only interpret. Next one. Yeah, next previous one. Yeah, here you can see this is the P00533. Okay, because in gene cards, you say that EGFR can be known by other names. So you need to know all those aliases, right? Whereas if you use this one, a single thing is sufficient because that one covers everything. Then if using gene symbol confirms organism and entry name, check protein length, download or record the exact accession. Okay, when did you download it and date because database interfaces and converts may evolve. Okay, and yeah, next. Okay, this protein identity, 3D model viewer, confidence colors, of course you can download. Okay, this PDB file you can download. And once you download, whatever you do is you put it to all the softwares that the first day, yesterday, he showed about docking and all those things, am I right? That one uh, you can do it, am I right? And do not download and use the model before checking identity, confidence, and biological evidence. That's the key message. Next one, madam. Okay, now this is a thing very high, often reliable for folded domain interpretation. 70 to 90 is okay. 50 to 70 low, less than 50. Okay. And PAE helps judge whether the relative positions or domains are reliable. Okay, because you imagine that how it is predicting, how it is predicting. Can anyone guess that? How this AlphaFold is predicting the structures? Okay, it's a pure chemistry. Protein means what? Amino acids. When two amino acids are next to each other, okay, depending upon the nature of the functional groups, either it may repel, or it may attract, or it may fold. That's all. Because this is only 20 amino acids. Those 20 amino acids, depending upon how they arrange in a sequence, it will come a 3D conformation. That's why if you look at the RFC PDB, there is more than 100,000 proteins. You imagine this simple 20 amino acids are creating so many proteins. Okay, so that is the thing you need to know. Okay, confidence source are not declaration, they define the level of trust. Of course, if you go more than 90, it's very high. Next one. Then step by step, this one is a beginner friendly. Okay, search EGFR, confirm the entry name, identify extracellular, transmembrane, or and intracellular kinase regions. This is important because we know that in the gene cards, we say that EGFR in a chromosomal location, it can present in any place, all right, in a cell. So we need to look at the extracellular, and of course, I think for EGFR there is a PDB structure you can check it. Okay, EGFR is a receptor kinase. AlphaFold can explain overall domain organization, but we need to always check the PDB. Okay, and of course, you can also change this. Okay, next one. Okay, and these are the practical applications. Okay, you can understand the target. You can develop a hypothesis. Of course, you can also write how the mutations interpret, and also you can build the model, and also you can also do this one. Okay, structural hypothesis generation plus experimental verification. Always you need to do the experimental verification. Do not use it blindly. Okay, that's very important. Claiming exact drug binding poses without ligand bound validation. Docking into low confidence or highly flexible regions. Okay, ignoring alternative confirmations and making it conclusions. Next one. Yeah, these are the some of the common mistakes that what the people do is, okay. Yeah, next one. And this one, before opening the AlphaFold, you have to define again the research question. You have to identify the correct identifier in the gene cards, and you need to know what is its biological relevance. Okay, and you check whether the experimental structure is available in the PDB or not. Okay, while you are viewing the model and you look at these colors and you look at avoid exact residue level claims in low confidence regions. Okay, because if you open the protein structure for each and every region, it will tell you the confidence levels. Okay, and take screenshots. Okay, only after adding interpretation, not as a evidence alone. Then after viewing, you compare with PDB structures, click literature, then record, then use the model. Okay, and combine AlphaFold, PDB, literature, and biochemical validation. Next. Then this is the minimum reporting checklist. Okay, all these things. Okay, protein name, gene symbol, everything. Okay, and example reporting sentence. Okay, this is what you need to say. The AlphaFold DB model for human UCFR. Okay, you see within the bracket you need to put the UniProt ID was inspected to understand overall domain architecture. Interpretation focused on structured regions with high PLDTT confidence. Ligand binding conclusions were not drawn from AlphaFold alone and were compared with inhibitor bound PDB structures wherever it is. So this is what you need to say. Okay. Why reporting matters means it improves the reproducibility, it prevents the overclaiming, helps reviewers, shows scientific maturity. Okay. Next one. Yeah. Now you can see that AlphaFold plus PDB, the Campbell, that what we are going to talk about. We are going to talk about PubChem, Swiss ADME. All these three are interrelated. All are interrelated. Okay, when it comes to the drug discovery point. So it will tell you the domain architecture. These are the experimental structures. This one will know the what are the reported compounds. These are tell you that whatever the compound that you are thinking, whether they are similar or not. And these are the ones that they will tell you that how toxic it is. Okay, then AlphaFold becomes more useful when connected to the orthogonal evidence. Next one. Yeah, this one. Start with a defined target question, use a reliable identifier, confirm organism, all these things. Okay. Of course, these are the workshops one. It is already there in the notes. Okay. Do not ask what structure can I give. Ask what decision will this structure support. Okay. Then, yeah. Next one. Yeah. AlphaFold is a highly valuable browser-based. Okay. Should be integrated with gene cards, PubMed, string. Prediction, interpretation, verification, and experimental planning is strongest when used by scientists who understand its limits. Actually, this particular statement is applicable for every AI tool. Okay, next one. I think this is the one. Next. So this is the one, and in AlphaFold, actually we did not give what I say is in the workshop where I put the AlphaFold and PDB together, okay? But I did not give a much emphasis on AlphaFold, but I give a much emphasis on the PDB so that you will understand more science. Then later on you can explore more on AlphaFold. And AlphaFold is the one we should really watch in near future because it may change the entire drug discovery. Okay. Thanks. That's the I think all the thing. Thank you. Okay, of course. Everyone says no, but I asked you, did you realize that one of the Nobel laureate, Nobel Prize winners is that guy? Okay, because uh, to me, that is the first instance where the AI is being respected by the scientists. Until now, in this part of the world, we see AI is very high. In other parts of the world, usually the people don't accept it. Okay, and but AlphaFold is the one where the people are talking about uh, is going to be game changer. So here the take home message now is uh, now you can discover medicines for any topic. That is the take home message with AlphaFold. Okay, that means what I say is you discover, but I'm not sure how many of them will be converted into medicines. That is still a big question mark. Okay, but it's okay. In another 10 years or 15 years, definitely it is going to be there. So the take home message, and also it is also one of the examples that you should be domain expert, then take the help of AI. Okay, don't make use of AI for your domain expertise. These are the key messages.
Thank you, sir, for your elaborate session. Now I request all the participants, both online and offline, to proceed for the lunch session. And the afternoon session will start at 1:30 p.m. and the live stream will be proceeded from there. Thank you everyone for joining this session. We'll end the stream.